Well, thank you. Thank you very much for the introduction, and thank you very much for the invitation to come and talk to you a little bit about my experience in trying to get information on what I believe are potentially interesting aspects of biology, which I've been working on for many years. Now, I would like sort of to start and share with you the experience that I very often had when I was discussing with fellow scientists, fellow researchers, the choice of topics that we're working on and the approaches that we're trying to adopt and use in order to do our science. And I believe that we all have certain, I would say, dimensional preference in the research in trying to choose the way in which we explore, in our case, biology. And my feeling, I think, all through was that it's very difficult to determine which problems in biology are more important than others. I think it's very much in the eyes of the beholder, and we all have different ways of trying to understand processes in biology, and to some extent with a dominant approach that is looking at having molecules that can be combined with specific processes by knockdown or overexpression experiments as an indication for understanding function. My feeling was that mostly the list of components was and is a secondary issue. It's a very convenient one in order to study function, but it's not really showing us or providing us information about the functionality, while looking at images and trying not only to see them and describe them, but understand the relationship to specific functional properties of cells, like division or migration, and so on and so forth, I think is bringing us very close to understanding processes. But in order to do that, we very often have to realize that cells are operating in a complex, fairly complex systems of interactions and not acting as individual elements, biological elements that are performing autonomously all the features of life, but our cells are living, especially in metazoans, are living in an environment that contains additional components. They contain a soluble medium around them. They contain some matrix to which they are non-cellular matrix, which is to which they are adhering, and there are neighboring cells which interact with them through different junctions. And all those interactions that cells have, face, perform, produce, have a huge role in directing the transcription of the cells and the division and survival and differentiation and the migration and invasion in some cases, and the remodeling of the matrix and so on and so forth. And some processes that are being provided by outside-in signaling, and there are inside-out signaling that change the matrix. And the number of components that are associated, molecules associated with adhesive processes, is very large, far beyond what one can assign to individual components, specific individual functions. And the type of signals that cells are getting from their environment can be roughly divided into two types: chemical signals, which are depending on the molecular composition of the matrix or neighboring cells, and thereby cells can make different types of adhesions with different elements in the surrounding. I'll give you a few examples for that. And there are physical signals which are less understood, less known and clear to us, and they are the topography in which the cells are growing, or the geometry of the matrix, or the density of ligands, and the rigidity and force, and so on. So cells cells are small, as we all know. Cells look sometimes like rather simple machines. In fact, they are capable of accepting and sensing a huge number of signals at any given time. They can integrate this information, they can read the information that they get from the environment, and they can make a decision in a way and respond to those signals by doing some specific actions. Now, all this scenario of interaction with the matrix is occurring in environments and with components of different sizes that span almost 8 or 9 orders of magnitude, from the macro view of tissues and organs and whole organisms, through which is exemplified here in this kind of section for the skin, down to looking at individual cells, then looking at areas of adhesion and individual adhesion with the matrix, and ultimately with molecules or fraction of or parts of the molecule, domain of molecules that determine the activity. In order to understand certain processes that have these manifestations at the macro scale, very often we have to go to one of those dimensional levels and study things in detail in order to understand how the cell is cooperating with the environment. So what I'll be trying to tell you in the next 40 minutes or so is in a kind of a very, very simple summary, that matrix adhesions are highly complex and diverse structures. Not all adhesions are carved from the same stone. They can be very different. They are also highly dynamic structures. They are not adhesions that are there forever or for even very long time. Molecules that participate in their formation are exchanging all the time, and they can transform very rapidly. In studying these structures, we need to have tools that enable us to quantify all these parameters, both the effect on the cells and the shapes and morphologies that are generated within the cells. We need to quantify them, which means that a major part of our work and our way to understanding the biology that we're interested in is through imaging and processing of imaging in order to get quantitative data. And some of these quantification of structures can go to nanoscopic substructures, as you will see later on during the talk. I'll also show you that adhesions in particular can be highly mechanosensitive structures within cells that are responding to mechanical cues imposed by the environment or by the cytoskeleton in individual cells. And I'll be trying to show to you towards the end of the talk, or at the end of the talk, that part of the force-dependent responsiveness of cells has to do with protein conformation switches that actually enable cells to sense, and I'll be offering you a kind of a model that provides a good example for that. So my story with this structure starts at the late 70s, when during studies on cytoskeletal components that are associated with membranes, I discovered in the lab of John Singer in La Jolla San Diego, the molecule called vinculin that is forming or that is associated with focal adhesions. And since I'm going to use this terminology a lot and not everybody here is familiar with the structures, let me just indicate that we have these adhesions, which are matrix adhesions, shown here at the bottom by a section in electron micrograph. You can see a kind of an elephant foot, which is associated with a dark line that shows the matrix. If you look carefully at that region, you'll find a lot of cross sections for filaments. The filaments, some of them are actin filaments with diameter of about 7 nanometers. Some are intermediate filaments that you can see here and there, which are about 10 nanometers. And around them, there are some microtubules. There's a network of such structures which is associated with the adhesion sites, primarily in areas kind of within the cell, while the area which is very much at the outer perimeter of the cell shows these initial adhesions that are called focal complexes that are still very poor in expression or association with actin. It just is starting to accumulate in these regions. Now, it turns out that from our knowledge that was accumulated over time, that the focal adhesions are made of specific components. I'll be talking about them in a minute, but there are many other structures which are associated with adhesion that are called fibrillar adhesions, which are derived from focal adhesions, are associated primarily with filaments of extracellular matrix like fibronectin filaments, which is a soft matrix outside the cell, and they have a slightly different composition from that of focal adhesions. I already mentioned the focal complexes that are located at the perimeter of the cell, really at the edge of the cell, and are containing most of the molecules that are associated with focal adhesions, perhaps except 2 or 3 that we know that joins in only later. And here you have a fourth example of an integrin adhesion that is forming a structure called podosome, in this case present in cells that are degrading bone, called osteoclasts. And you can see the structure, which looks very different from focal adhesions. And in the middle, there should be actin, and the perimeter, there's vinculin, and a few additional molecules of the same family. And all those are very much the same, almost the same in all the adhesions, although the different adhesions have totally different functionalities. And you can get here an example that even within the same cell, if we are trying to do what we call ratio imaging, namely we are double labeling a cell for 2 components, in this case, to phosphotyrosine and a molecule called zixin, and we are now calculating for every pixel in the image. We are calculating the intensity of both of them, and we calculate the ratio between the 2. We can see that there are regions that contain both molecules that show here as kind of yellow or greenish structures. We have phosphotyrosine containing structures which are only red, and we have a lot of areas with zixin that do not have phosphotyrosine in other regions which are primarily blue. So the focal adhesions that we are looking at are looking very much the same or very similar, but we can see that even within the individual focal adhesions, different regions may have different compositions with regards in this particular case to the 2 molecules that we've been looking at. Now, you've seen the example here of 2 molecules that were stained that shows you some of the part of the complexity, but really to appreciate the overall complexity, we know today of over 200 molecules that seem to participate in focal adhesion formation. The basic component, the receptors for the adhesion, are integrin molecules that I'll be talking about in a minute. There are additional membrane molecules that are working along with the integrin molecules, but in addition to those, you have many other molecules that either form a scaffold that connects the receptor eventually with actin filaments which are part of the cytoskeleton of the cell. And there are many additional molecules that belong to different functional families that are summarized in this simplifying slide, in which you can see that the components associated with adhesion sites in what we call the adhesion. Many of them are regulatory molecules like lipids or kinases or phosphatases or GAPs, proteases, chaperones, and others. And a few are scaffolding molecules like actin itself and the adhesion receptor and adapter proteins that connect the adhesion receptor to actin and to actin regulators, as you can see them here. So this is a scaffold, and these all are regulating it by phosphorylating some components or dephosphorylating some components, and by acting as stimulatory lipids that affect the behavior of the molecules. So the system is an adhesive system. It has the capacity to support cell adhesion. And as you'll see later, cell adhesion will be necessary for many, many other processes within the cell. But if we want to understand how focal adhesions actually work, we need to know a few things. We need to know their nanoarchitecture, for which we need to develop multi-scale and multimodal imaging technologies that combine, for example, live cell fluorescence imaging and the cryo-electron tomography. And that was one of the early studies that we conducted over 10 years ago with Ohad's group. The main finding is based on correlative microscopy, which is not exactly trivial, which is correlating focal adhesion that can be identified by the focal adhesion molecule, which is tagged by a intrinsically tagged by a fluorophore that enables us to look at the adhesion sites, for example, the one which is located over here. And then the cell, after taking such pictures, in this case of a fixed cell, looking at the cryo-electron tomography from different angles. In this case, this part of the work was done primarily by Ohad's group. And by looking at the structure, one could see the cytoskeletal components, which are rendered over here. Sorry, we can see here some vesicles that are apparently located in that region. And we see interesting structures that are these bagel-shaped or donut-shaped particles that in higher magnification have this kind of appearance associated with actin filaments. And we were trying to figure out what they contain. We don't know exactly yet what is the composition, but we do know that in terms of the organization, these structures are organized in layers where we have the membrane, we have these donuts, we have actin filaments emanating from the donuts, and then we have, to the deeper area within the cell, we have the actin filaments, a cable of actin, parallel actin filaments. And that was, I think, an important collaboration that started a whole series of other interactions in which we try to span this huge difference in resolution and in detail and dimensionality between the adhesion, what's happening in a living cell, and what one can visualize looking at cryo-electron tomograph. And you can see here some very preliminary at the time examples of these donuts as they appear in the cryotomograph, and you can see next to them labeling with gold labeling for vinculin, in which we compromised a little bit about the structure, but we could demonstrate that vinculin as a molecule can be immunolabeled in the vicinity of these donut-shaped structures. So fine. So one issue is that we regard, in a way, adhesion sites, especially adhesion to the matrix, as structures that are essential for tissue formation, for linking to the extracellular matrix, sometimes to neighboring cells, very similar type of structures. But in fact, adhesions are affecting not only tissue formation and remodeling, but also are critical for cell motility. They create a kind of contact that enables the cell to hold on the molecule on the matrix and then pull itself along and move its different parts. Obviously, this can also serve for cell invasion, mainly in metastatic cancer. The adhesions to the matrix can reorganize or induce reorganization of cytoskeletal components, and they can guide migration in a specific manner. And also via the same signaling molecule that I showed you a few minutes ago, the adhesions can form and support cell survival by affecting the signaling cascade of processes that occur within the cells. Just to illustrate to you, and I'm trying to focus a bit on technologies that were used, that we've been using throughout these studies. I can show you an example of how the adhesions are critical in migratory activity, in which we can control or regulate the adhesiveness of the cells. And what you can see over here is our cells that were plated on a matrix that was covered with a very, very special coating, which we've been working on with the Joachim Spatz laboratory for quite a number of years. So what was done here, I mean, we have coated it, the surface, with a material that is actually anti-adhesive. That is anti-adhesive, and then we shine light on this surface with a special configuration that can, which can switch the substrate from being non-adhesive into adhesive by cleaving the repelling part of the molecules in the coat. And then, as you'll see in a minute, you'll see that these cells that were confined to interact at that stage only to the adhesive kind of bagel-shaped structure, and they try to move around. And you can see this, this is the UV flood, and then you can see that the cells start to move away from the initial location. If you watch it carefully, you can see that they are dividing, which they stopped dividing when they could not expand and move because they were limited in space. So there is a migration which we can, for example, temporarily control by photoactivation of substrate, or we can work with a different type of system for monitoring migration, which are substrates that are covered with small beads that turn the entire surface to look in the microscope, in the phase microscope, for example, to look gray. And then we can plate cells on this layer, and the cells, when the cell starts to move, the cells will start to push aside these beads and create tracks that can be identified, which you can follow in snapshots at the bottom here, and you can see them formed by two cells over here. And from looking at these tracks, which are providing the kind of historical appearance of the movement of the cell, which is picked up here in specific snapshots, can enable us to calculate a whole variety of parameters using methodologies that were developed mainly with Ofra Golani at the Weizmann Institute, and enabled to do this quantification, which can enable us to distinguish such a track, for example, from such a track. This one is made by a specific non-small cell carcinoma, H1299, in this case melanoma. And just to illustrate this to you, you can see here an example, part of work of Suanafar Abu Amara, was working here years back, that can demonstrate that the cells are quite nicely migratory, as you can see over here. And by knocking down specific gene, one of the variants of PKC, protein kinase C, we lose the capacity of the cells to form these elongated trajectories, and they are primarily chewing or moving or pushing aside a little bit the beads that are located next to them. And what Ofra managed to do was to segment these structures, identify the structures that are suitable for measurement, and then define their different parameters like area and axial ratio and so on, which can be provide us with functional information about the adhesion. Okay, so we discussed the fact that adhesions can come in different flavors, different shapes, and they have different functions, even if they're coming or formed by the same cell on similar substrates. Then we have discussed the fact that the adhesions are dynamic and they are driving and important for cell migration. And we have discussed the fact that all those processes or behaviors that are depending on the adhesion migration of a cell can be recorded and can be quantified by processing of images in a way which is precise and relevant for the functionality of the cells. Now, the cells, the interaction of cells with the extracellular matrix that is underlying them, located on the substrate, are capable, as I already mentioned, to display both chemical and physical diversity. Matrix components, for example, can be recognized by specific adhesion receptors, and a common family of such receptors, which we and others have worked a lot with, is the integrin family. It contains sort of two chains that cross the membrane and interact with the extracellular matrix on the outside part of the cell. And there are different combinations of alpha and beta chains that build this heterodimers, over 20 variations, each of which has its own specificity. So the cells have receptors. The receptors can identify or see differences between two different ligands of integrin, which are fibronectin and vitronectin. You'll see it in a minute. And there are also a lot, but still very poorly characterized, adhesive components outside cells, which are not interacting with integrins, but with many other molecules. I'm just mentioning it because the presence of a vast information about integrin adhesions and about fibronectin, for example, people sometimes forget that the diversity that the cell has in order to face the matrix and interact with it is much, much, much broader. So just to give you an illustration of that, I think this is from studies that were carried out at the time by Baruch Zimmerman, and some studies along that line were carried out by Tova Volberg, who worked with us for many, many years. And you can see here an example where we allow cells exactly the same cells to interact with two matrices. One of them is a matrix that contain fibronectin, in which we also block, in addition, the potential to interact with vitronectin. And we have now surfaces that are coated with vitronectin, and we block completely the interaction with fibronectin. And as you can see, without doing any measurement, just looking at the images, you look here labeling for 4 different molecules of focal adhesions. And I believe that you can see in this case that there are a lot of adhesions throughout the entire cell, at the periphery of the cell, a lot more in the center of the cell. And the cell is very spread, while the ones that are interacting only with vitronectin have a few adhesions in the center, but they are not very developed, and most of the adhesions are at the periphery. And we'll get later towards the end of my talk, you'll see an interesting example of how this can be related to a very, very specific feature of this molecule that we like so much, vinculin. Okay, so that's what, one example. Here you see the scheme of different integrin and combinations, an indication with what molecules they are interacting. The details here are not very important. And here I can show you an example from a study carried out recently, very recently, by Wenhong Lee, a graduate student in our lab, that she looked on cells that are interacting with fibronectin and with a molecule called galectin-8. Galectin is a lectin. It's an animal lectin that we have coated the surface with. And you can appreciate, I believe, we are labeling here for the cells for actin and for myosin, as you can, I think, myosin 2, as I think you can appreciate. And you can see that this cell has a much stronger tendency to spread. It spreads very strongly on galectin, much stronger than fibronectin. And fibronectin, on the other hand, you can see that the cells is spreading quite a bit, but it forms a lot of very, very significant arrays or bundles of actin that are actually contractile, and they are limiting the spreading of a cell. And just to illustrate that to you, I would like to show you a movie that demonstrates by a technique called this, by optics that are called interference reflection optics, that is enabling us to measure the distance between the bottom, the ventral part of a cell, and in this case, the surface glass on which a cell is spreading. And you can see here different time of spreading on fibronectin, on galectin. You can see the differences. And I believe you can see here the process of the spreading, which is quite interesting, in which a cell is pushing out lamellipodia, which are these structures, flat structures, and filopodia, which are this finger-like structure at the cell periphery. And if you look carefully at the edge of a cell, you can see that the process is highly dynamic, with kind of a flow of material from the periphery towards the cell center. This is the area which is full with myosin that you saw before. And you can see the structures which we have characterized, or Wenhong characterized quite extensively, demonstrating, for example, that these filopodia, these finger-like structures that are being extended by the cell, they contain a very specific type of myosin called myosin 10 at their tip, which is promoting the extension growth, and even doing something very, very similar, which is having a tendency to turn always in the same direction in a chiral fashion. This is kind of a turning, which is a left-handed turning that is exhibited by many of the filopodia of this particular cell. In this case, a cell contains GFP-tagged myosin 10, which is demonstrating this kind of distribution. Well, I will not go in great detail into the entire process, but of formation of focal adhesion, just to let you know that it starts with the edge of a cell when small dots are being formed. As you can see over here, this area is exposed to flow of actin from the periphery towards the cell center. And when it goes over these dots of initial adhesions or focal complexes, they start to connect to it. And I think you'll see another example of our collaboration of Ohad and our lab in how this is actually taking place. Now, the next is, since I believe that I need to rush a little bit, we lost some time at the beginning, so I'll try just to go quickly through one feature which I believe is very important for us to identify, which is the capacity of cells to sense through their adhesions physical properties of the matrix, like topography, geometry, density, agility, and applied force. And this kind of phenomenon was found by Daniel Rivlin, who was working with Sasha Bershatsky and myself as a postdoc. And the demonstration that Daniel showed was that if you take a cell that contains fluorescently tagged focal adhesions with a variety of components of focal adhesions, and you pull on it with a pipette, so this area feels tension. You can look at this particular region. You can see that the adhesions that initially were relatively small, they grow considerably, and they are become full of actin and full of vinculin in this particular case. And this is in a way unexpected, at least superficially, because you would expect that if you are pulling on two elements that are linked to each other or adhering to each other, you may break the adhesions, but that's not what's happening unless you really employ a very, very high level of force. But it is actually quite expected functionally, because adhesions are made in order to keep cells attached to a surface. And apparently, cells have developed a process that when you are pulling on it from the outside, by either stretching the cell or doing this trick with a pipette, the cell has a way to compensate for that by increasing the adhesion, and we'll try to get towards the end an indication how this is happening. So just to give you kind of an illustration of a summary slide from a joint study of Raja Patersky and Ohad Medalia, looking at the events that occur at the cell edge when the cell is expanding or spreading. There is actin which is polymerizing and in a way streaming towards the cell center, and on its way, this actin is applying force to initial molecules that are associated with the early formed integrin adhesions. And when this is happening, something is happening to these molecules. They become activated. And we'll see a bit in a few minutes how this activation actually takes place. The form of the activation is primarily by conformational change, that the molecules, if you can just watch the arrow, are transformed from this kind of tail to head link that is keeping many of the binding sites of the molecule to the inside or unexposed to the environment within the cell. And you can see that at certain points, there is a spreading both of tailin and vinculin that can then link to actin. And when they link to actin, they are bundling actin and forming very, very dense bundles. So this was a very important observation published about 2 years ago. And it suggested to us that some of the dynamic processes within the cell, like actin polymerization, might be effectively changing or activating molecules to open up and become active and expose their business part to neighboring molecules, and thereby organize a cytoskeleton in these beautiful structures. So we wondered how this is happening, And Dror Chorev, who was working at the time in collaboration with Michal Sharon, who is a specialist in mass spectrometry of complexes of different types of molecules, carried out an experiment which was based on checking how much force or energy do you have to apply to, for example, a vinculin molecule in order for the vinculin to open up. And that was done by a methodology that is defined as kind of force-dependent clashing of molecules onto surfaces, and then enabling us to actually measure the surface area of the molecule. And you can all understand that when the molecule is sort of opening, the surface area increases, and when the surface area increases, it can give us an indication to the level of opening, and we figured out at that time that the molecule in the unperturbed vinculin is in the closed conformation, and then it is getting sort of into a semi-open conformation when the force or the energy that is invested in perturbing the molecule is increased, and eventually it is opening up completely and changing dramatically its experiment, the value that is derived from this collision based opening and unfolding of the molecule. So we knew that we can do it. We knew that that by investing energy in such molecule, we can open it, and we've sort of contemplated together with few other people, kind of a model that demonstrates what are the steps, major steps that take place when focal adhesions are formed. And the first is an interaction with the membrane of a molecule called taline. Taline is a vinculin binding molecule, which is in a way which is folded quite strongly when it's in the cytoplasm. And when it interacts with the membrane, it starts to loosen up and open up, including exposure of a specific domain on the molecule, which is what we call the vinculin binding structure or segment of the molecule, the VBS1 in this case, which is located along the rod of taline, become exposed. And then this region is confronting with vinculin in its folded state, and vinculin at that time starts to open up. And when it's opening up, as you can see over here, it opens its tail, and its tail catches actin and cross-link actin in a way that I demonstrated to you in the one of the previous slides. And here we actually came to a discussion and some idea that developed with our collaborators in Heidelberg, that have studied in detail the changes within the vinculin molecule that occur when it interacts with this VBS1 molecule. And you can see them here, Frauke Greter and Florian Franz, and they actually doing analysis of conformation analysis based on forces and levels of energy within the molecule. They predicted the change that occurs in the vinculin molecule when the VBS is interacting with it. And when we discussed this issue with them, we, in this case, was Oad Medalia and myself, we asked them whether it would be possible to try to see whether they could activate vinculin not without the VBS1 by simply making or creating mutations in the vinculin molecule that will, in a way, push the molecule to change their conformation and change their folding capacity and be very similar to VBS-treated vinculins. And they predicted for 4 mutations, of suggested 4 or 5 mutations. And the first thing that was done was checking this molecule, which was done by Ohad. And he could demonstrate that when you take vinculin without the VBS domain, and you can get a certain bundling, level of bundling of actin, but in the presence of the 4 mutations or the 5 mutations, there was major increase in the bundle formation, and by adding VBS1 plus the mutated vinculin, they could get a very, very similar effect that augments the formation of the structure. And we did find, interestingly, that vinculin is needed for integrin to join into focal adhesions, which was actually not clear from the start. And by looking at the focal adhesions formed by the wild type compared to the 4 mutants and 5 mutants, we could see a very different form of adhesions that were generated within the cell. Primarily, we saw that the cells with the mutants have formed low tension central adhesions that are formed by the mutant, but not by the vinculin, that requires a lot more force in order to be activated and to form adhesions. So this is just a quantification of the images that we have just showed you, demonstrate here that, that when we look at central versus total number of adhesions, the vinculin itself is very ineffective in making central adhesions, and the mutants are very effective in doing so. So that's what I wanted to discuss with you and share with you. I sort of selected for this talk a variety of approaches that are asking and addressing a major question that is associated with the functionality and the way of formation of focal adhesions and their capacity to act as surface sensors. That is, of course, only a small part of the work that was carried out in the lab throughout all these years, but I selected here a few that demonstrate the capacity of research in that field and analysis of the images that are being formed by different forms of microscopy to figure out how changes that are ranging from effects that occur by conformational changes in individual molecules and go through formation of clusters and continue with formation of cell clusters and tissues to realize that this system is of great complexity, diversity, and most critical for the large number of functionalities of different cells. I tried to mention contributions to these experiments of different people in the lab. These are people who were in recent period in the lab and that I refer to their work throughout my talk. And I'm also very grateful for many collaborators with whom we worked over the years with an attempt to get this kind of unique information. So I'd like to close here, and if there are any questions, I'll be very happy to answer. Thank you very much.